The question mark up here is about all sorts of questions. And the main question I'm going to start with is what I'm going to say to you relevant at all to your work? I would like it to be. And I'd like you to give me the opportunity to make the case that what I do is relevant. And we'll see if it works or not. We are all, I think, involved with representations. In fact, the New York Times on today's cover is the top fold, which is, as you know, the most important piece, is a representation. This happens to be about the cosmos. Back in the day, you know, these astronomers get a lot of attention, and that's one of my points why. I mean, that's a whole conversation, but people love looking at astronomical images. And we have to find ways of getting them to love looking at our work, and that might be a whole different seminar. But we are together talking about representation. For me, and this is where the question mark is, will my explanatory representations have anything to do with what you do? Because I believe that most of yours are exploratory. But I'm firmly convinced that we are all in this together, and that the idea of making a good representation needs good design. And that's sort of where I'm going to be heading, although I'd like to think other ways, other areas as well. Remind ourselves, again, this is so obvious that you say, why am I bothering to say this? But we lose sight of it because we get so involved in our own little tree in the forest. Remind ourselves that representations are representations. They are not the thing. The photograph is not the thing. The data is sort of the thing, but the representation of the data is not the thing. And so therefore, we as representers have some translation that we have to get involved with. We have to interpret. Ultimately, we have to make decisions. Once again, I'm not saying anything you don't know, but we have to keep that in mind, because I think most of us, including myself, we can get lost in our exercise and not pay attention to what we're doing. Ultimately, as I'm speaking to the choir, when we think about making a representation, we are clarifying the work for ourselves. It's not only about communicating to others, but the very process of making that representation, making those decisions, clarifies all that in our own heads. That, to me, is very exciting. That's one of the little secrets that it's no longer a secret, but I work actually all over campus, mostly in 4 departments, and the wonderful piece is that I'm learning. As I'm discussing how to represent something, it's a constant back and forth of talking about abstraction, and we'll get a little bit into this. But this is not trivial. In fact, I feel, and I'm not going to be able to get into this today, and I'm glad, Bang, that you brought up picturing to learn. I believe that this whole notion of making representation should be part of the curriculum. There should be visual literacy in all campuses. We should teach not only making representations, but when we look at something like this, people should understand what it is that they're seeing. Why is it colored this way? Does the color mean anything? All that kind of thing. We think about this, but not enough is happening in education. And I'm concerned, especially with the whole MOOC thing. I went to a seminar the other day, and they're not thinking about visual. And maybe some of you would like to talk about it. Okay, so when I talk to researchers about how I think they should think about making their graphics or images. The first thing that I think, again, my thing is explanatory. But I do think that in exploratory work, you want someone to use your tool. So make me look. And that it's actually not easy. There are basically 2 points I will be starting with here. And the second one is make me understand. Again, very simple ideas, but we forget when we're so involved. Making me look is different from making me understand. And the idea is how to first engage and then to ultimately get people to ask questions, and maybe they'll understand better. So starting with Make Me Look, I'm going to do a little plug for a book that I did with George Whiteside called No Small Matter. It's about science on the nanoscale. Very, very difficult to explain. It's crazy, that whole world of quantum mechanics and all sorts of things. I'll just quickly run through some images that I made for the book. This is about nanoscale. It's a drop of water. Any photographers here, by the way? Okay, just a couple. You might be, if you look really carefully at that drop, it actually acts as a lens, and you can, in fact, see the background becoming in focus. The background, I wish it were darker. No? See, photographers are control freaks. That's okay, if possible. But the background is something I bought a long time ago. It is a grid of pastels, colored pastels, and I knew someday I would use it. So when you use macro photography, you know, the depth of field is very limited, so the background is completely out of focus. But if you look at the drop, you could see the background. But this is about nanoscience. A drop is about molecular structure. What is going on at that boundary? What is happening? And I can't explain it in a picture, but George talks about it. By the way, all of the work that I do in photography is always accompanied by text. I am not an artist. I want you to understand the science of what you're looking at. Here we were talking about bacillus. We were talking about electron resonance, pushing the ideas a little bit. Again, what I'm getting at is making you look first. These are about bubbles. This is called, I don't necessarily name my images, but this is it's called Eleanor Rigby. Does anyone want to take a guess at what you're looking at? You're all too young. Yes! Somebody my age. It is in fact the vinyl album of that. I think it was Rubber Soul, I forget, of the whole collection. But it is in fact that track. And that is about nanoscience, believe it or not, and how sound is produced from those tracks. And I'm not explaining it in the picture, but I hope that I'm making you look so then you could then read the text that George wrote. Here's not a great picture that I didn't make. I took it off of the web. It's material, some of you might be familiar with some of this. It's called Ferrofluid. And this is the picture that I made years ago. I still show it because it's making a point. I believe that you want to see what, you want to look at this and ask questions. I mean, I'd like to think that that's why it's successful. Ferrofluid is material, it's actually a drop of oil that has suspended in it iron particles. That drop is about 3 centimeters across. So, you know when you look at iron filings and you put a magnet next to it, you get these feathery things going on. It's quite lovely. Well, this is the same thing that's going on, except this drop is on a glass slide. Under the glass slide is a yellow Post-it, very high-tech, And under the yellow post-it are 7 circular magnets. So you're seeing the ferrofluid respond to the magnets. This is a straight shot. This was an excuse for us to talk about magnetism. If you look carefully, you could see some green, which it's so highly reflective that I decided to put a green card over it. You could see the window pane. It was just ambient light with window. And if you really look carefully, you could see the sign of a very bad photographer, because I'm in there, which I didn't see until it was published. Here I am. That's the lens. You're seeing 2 lenses. This is my head. Actually, we use this for a magazine article for kids. Find Felice. But it's fun. I mean, the idea is that you want to look, and then you want to ask questions about what you're looking at. This is a story about hydrophobicity. That leaf is highly hydrophobic. And look at it's called the Lotus Effect. It's not a lotus leaf, it's another kind of leaf. But it is so hydrophobic that the water drop has become a ball. And again, it's our excuse to talk about stuff like that. This is a lousy picture that I made quite a while ago. It's actually a piece of a syringe that has embedded in it chips. The idea is to be able to take that syringe on site and do some blood analysis. Just look carefully as I simply change the lighting. This is a microscopic image, low magnification. Look at this. It's the same spot. And it's just a different decision that I made. And also, the background is different. But it's about trying to get you to pay attention and to look. Here's another decision that I made. This is, they're not stents. They look like stents. But Rebecca Jackman coated very thin glass rods, about 4 millimeters across, deposited metal, and then dissolved away, and the glass is gone. So you're seeing sort of stent-like constructions. And so I made this picture, and it's okay. This is with transmitted light. And here it is with epi-illuminated light. It's a whole different image. Because, first of all, I think it's more attractive. But also, you're seeing the 3-dimensionality of it because I'm including shadows. I'm not sure any of this is immediately relevant to what you do. But I think that if you can sort of take a step back, I mean, I would love someday for there to be time for me to make a case that it is relevant to what you do. It's about figuring out how to best present ideas and to not just stay with the first idea. I mean, again, that's another thing that researchers tend to do. They just make the graphic and it's good enough. I suggest we go beyond good enough. John Rogers made a wonderful image of his circuitry on plastic, flexible plastic. And he's showing a sense of scale. He shows it's flexible. It's good. It's a good documentary image. This is the one that I made that got on the cover of PNAS. I'm not saying it's better. It's different. And maybe you want to look at it further. Maybe. Maybe not. Decision-making in the works all the time. Just by changing the light source and the direction, the entire image changes and what you're seeing changes. Again, I think that a lot of you already do this, but a lot of you don't. And that's what I'm encouraging, to go beyond your first idea. Here's another lousy image that I made of a micro rotor that measures about 1 centimeter across. I had gotten a new, at that point, it was a fancy dissecting microscope. It's called a dissecting microscope. And the issue about it is that you don't have control over the direction of the light. So this micro rotor that goes into another engineering feat, you can see all this yuck and dust and scratches and blah, blah. So I just went back to my camera and lens. And it's the identical sample. I'm doing nothing to it. All I'm doing is using simpler equipment, which I have more control over. That's one of the things that photographers, as I mentioned, like to consider having more of. That's sometimes actually most of the times now we have very little control. But a decision, again, I made a decision that even though that was a cool piece of equipment, it didn't do the job. This is an image coming from the lab. The researcher made the lab of a device. And this is the picture that I made of the identical device. First of all, I got it in focus. and all sorts of ideas, small ideas, yes, they're not profound. They're not about deep science. But the decisions that I made, I think, made a better picture. This is about lighting and background and angle and a lot of other things. And I know that your work is about that as well. I got some microfluidic devices in the mail from Steve Quake a while ago, and this is ultimately the picture that I made of it. Jerry Fink's yeast colonies, I lit one way, and I lit it another way. It's the identical composition, if you could see. Once again, I'm not sure which is better. I give that choice to the researchers. But it's about making decisions about things. In photography, it's about light. Here were cuvettes of something called nanocrystals. They're cadmium selenide. I put them in cuvettes and just made a stupid image, which I had fun with. But this is, in my opinion, the better picture. What's going on here is these nanocrystals, these vials have the identical material. The only difference is the size of the nanocrystals. So when you excite it with UV light, it fluoresces at different wavelengths. So that's the point of this science, that they're creating this material that can fluoresce at different wavelengths. But just by looking at this, which I think is a successful picture, and by the way, which a lot of people steal, but that's another conversation. Can you tell me where the camera is? How did I make it? This is not a test. This is just.
From above?
Yes. How do you know it's from above?
The bubbles.
Thank you. The bubbles. I thought that was a sort of a subtle, nice addition. Well, they didn't like it. This is the picture that they wanted me to make. Straight on, and it got on the cover of an important journal. Eventually, they came around. But you know, this kind of decision making sometimes works and sometimes doesn't. It depends upon how the researchers trust me. They're starting to after 20 years, they're finally trusting me a little more. I show this a few times because it's something that we really need to think about. Even though this is about a photograph, it's about everything, really. This is another image from Jerry Fink's lab. It's a yeast colony. It's positively stunning. Now look carefully at the screen as I digitally delete the Petri dish and the background. I'm going to do it again. This is a beautiful, beautiful thing. The morphology has not changed. I've kept the science, in my opinion. Jerry felt that I deleted information about the size of the yeast colony. I suggest that you could say that in the caption. I want you to look. That's what this portion of the talk is about. I want you to look at this. Look what's going on inside. I mean, it's amazing. It's positive. I have the best job in the world. But I made a decision about deleting that background. And so the point of that is that, yes, I'm making you look, but I believe that I'm being responsible in doing what I do. By the way, anything that I do in any image, and sometimes I do color SEMs, for example, I always indicate that I've done so. It's critical. Again, that removes me from the whole art world. You have to know my process. This image, another representation, is dead wrong. And it's a very, very powerful image that the public has imprinted in their minds about the story of evolution. This is an irresponsible representation. Throw out some ideas about why this is wrong.
It doesn't branch.
Great, it doesn't branch. What else?
Too proscriptive.
Sorry?
It's too linear.
It's too linear. Of course, absolutely. It's too perfectly spaced. We do not all become male, nor do we become white males. And the most important thing, and I showed this to a group of science center directors, it doesn't show extinction, which is the critical part of evolution, which is what you suggested about the branching, but even further. That is the story of evolution. So this whole idea of representing ideas, these ideas get to the public, and the public, the power of images is extraordinary with the public. We have to be responsible. We have to think about this stuff. This image, well, what we're getting at is the notion of responsibility. How many of you have seen this image before? Okay, great. So you know that this is an image that was made from 100 other images. This is a composite done by David DeRosier. And maybe I'm going to be saying something that you already know. But I want to read to you what the journal indicated as in the caption, that the motor proteins do not survive the extraction procedure, and so are not shown. So there is a level of uncertainty in this image. It's not as perfect as, I mean, it's pretty amazing. It is a E. coli motor, a flagella motor. It's mind-blowing that they got such, what seems to be such precision. But there's stuff that we're not showing here. But unfortunately, and therefore, in my opinion, we have to say that somewhere, that this is not, once again, the thing. The problem is that when you see something as cool as that, and what is so possibly as perfect as that, you run into the problem of getting people who love making illustration to extract the idea and make it into this really cool looking thing, which eventually can be used to support intelligent design. Because the fact they say that intelligent design is a better explanation of something so incredibly perfect than chance. This is what I'm getting at about being responsible when we publish anything. Somewhere we have to say, whether it's data, if your graphs, your whatever, your images, that this is a representation, getting back to the idea from the very beginning. It is not this. So getting back once again to the first point, making you me look, I really am convinced that talking about our process of how we make representations is a very interesting idea. People are interested in how you think. And so, for example, I give this example where in the book I wanted to make an image of a nanotube. Now, there are other people who've done a much better job using their SEMs making beautiful images of nanotubes. What I wanted to do for the book is to, make it a little more interesting to look at. So all I did was I imprinted on acetate, that's what you're seeing, an 8.5 by 11 piece of acetate imprinted with the hexagonal graphene pattern. I then took that, you have to use your imagination, pretend this is the picture, the paper. I then made a cylinder out of it. Now, it turns out that when I had to make this cylinder, I had to figure out how to connect the edges. In the making of this image, I had to start thinking about how I was going to do it. And in reality, depending upon how you attach the edges, you make different configurations of the nanotubes, which then affects the conductivity of the nanotubes. So I'm learning about the science as I'm making the image. Then George said, let's start, let's kind of make the carbons a little more active, and this is pretty pukey that I did, and then we knew that was not going to work. Then maybe we should make the electron clouds. We should sort of indicate the electron density, and that was even worse. But once again, this notion of making a representation, should I make the density clouds this way? Should I make them that way? I don't know. Making a physical representation that's something that's not physical is something that is actually very exciting to think about. And that kind of discussion has to go on, not just by myself, but among the researchers and students as well. Finally, I wound up, just doing this, and in the book, this is what we wound up with, a little Photoshop and, here and there. But again, this is a book for the public, and this is our way of engaging them to understand what a nanotube is. The second point is about making me understand. After you look, let me try to get you to understand. For the book that we did, again, this is photography, and this is probably not relevant to your work, but I'm going to show you anyhow because it's fun. It's using metaphors is a very, very powerful idea, in my opinion, in showing, in giving people a handle of where to even begin. But in the process of thinking about those metaphors, it clarified my thinking and George's thinking. We had this conversation about whether the metaphor works, maybe it doesn't work. This is a metaphor for etching with light. Binary, how computers read. It's an inside of a music box. It goes on and off. When we wanted to talk about how an atomic force microscope works, I made an SEM of a tip, and then used pin art now to sort of suggest the rastering of the AFM tip, and that it sort of senses the substrate. Have any of you know what I mean by pin art? These are hundreds of pins that when you put something under it, it reads it's feeling it. It's not perfect, but it gives you some sort of handle on how an AFM works. Electron flow from one orbit to another, maybe not. The whole notion of metaphor is very, again, powerful, and I believe very accessible. This is one that I showed a while ago that I did delete just for a moment the text so that you could see the image. And this is how the image was used. It's right after 9/11, showing. Now, we could talk about where it falls apart and, how effective it is, but I do believe that metaphors are another, remember, they are representations and powerful. Here's a piece of one. It's a third of an image. This is the whole image. By the way, I showed this recently, and somebody came over to me after the talk and said, "You really shouldn't show that." Guess what country he was from? No, that's not fair. Although, you want to guess? What? All right, never mind. Okay. But you get, but better, people get it. People get it. Actually, they enjoy looking at it. This is not the metaphor. This is the information of the universe. Here is the Fermilab's metaphor. I think that's, I'll go back. This is showing dark energy and dark matter, and I think it works. It's not deep, it's not quantitative, true, but it gives you an idea of the dispersion in all this. So, again, I'm trying to make you understand, but, and here's where I think a lot of us fall apart. To get you to understand what is it you want me to see, because that we are so involved with our own work that we just make the assumption that people see what we want them to see. And Angela DePace and I wrote this book because of, I think, this challenge in the laboratory. We call it Visual Strategies, a practical guide. What do we call it? A practical guide to graphics for scientists and engineers. It's, by the way, it was designed by Stefan Sagmeister, who's a very prominent graphic designer. That wasn't an accident. We truly believe that graphics people should be part of the conversation. In fact, I think that should be a course, but that's once again another piece. So, okay, so for example, Don Eigler's Quantum Corral from years ago. This is their remember that we're looking at numbers that have been translated into an image. And some computer scientists decided that these are the colors that they wanted to show. It turns out that this Quantum Corral, which you probably, how many of you have seen this? Okay, good. So these are atoms that have been literally placed on a substrate in a form. I mean, they had that kind of control. Within the corral, there are quantum effects going on. I can't say anymore, I don't understand what that means. But I do know that the quantum effects are very, very important in this image. And Angela and I are suggesting that if, in fact, those quantum effects are just as important as the corral, then maybe it's better in grayscale. We could talk about this, and that's what we're trying to do in the book. This is how it appears. We have a lot of befores and afters showing how the image or figure appears in the various journals, and then we have our ideas, and we sort of we tell you over here why we've suggested it. George Whiteside's, again, made this image for actually a submission. They wanted to show off all the amazing devices that they were able to make. And I have no idea how to even look at this. So what we're suggesting in the book is put some sort of order to this. Let's talk about composition, for goodness sake. You know, for us, we decided to actually go back to color, where some of the images, I didn't make these images, that people took it in color, put on a different background, and order it in scale. And then perhaps instead of sticking all these letters all over the place, perhaps it's better if you have a legend like that. Again, I'm not saying this is the way to do it. What we're trying to do is get this kind of design conversation going. Once again, these are explanatory images, but yet we're talking about design. And I think that you also are talking about design. And there is a connection. I hope that you agree with that. Here's another example where the researcher, this is two kinds of explosions. They want us to see this area. They want us to compare this and this. And we're just saying, okay, you want us to look at those two? Get rid of the stuff that prevents us from seeing what you want me to see. Again, it is a problem. We all have that. I mean, I have it too. When I work for hours on a particular picture, I can't see it anymore. And so it's critical to work together, have a fresh eye come in and really make suggestions, which is what I think you probably are doing here. And this is what we started doing a while ago. Here's another one. This is from Science, where this was submitted to Science by a researcher, and this is what Catherine at Science simplified that submission. This is a submission, you know, it shows some sort of cycle and how of a particular species, and how weather and water content and all kinds of things. I mean, I have no idea how to look at this. So Glenda made this out of it. It's true you have to, this is annual reviews. You have to look a little carefully, but I think it's like night and day myself. And we have all these in the book. This is an original image from the researcher. What they're doing is, how are we doing with time, Bang? I don't, because we lost some time. I'm going to try to hurry. Say again? 10 minutes. Yeah, I better hurry up. Okay. So this is from the researcher showing the difference between an aging brain and an Alzheimer's brain. But this is not brain activity. It's not fMRIs. This is thinning of the brain. So it's a physical difference. We're showing physical differences. However, what they're doing is using colors from fMRIs. So I asked the researcher, if you want to show thinning, why are you using the color palette of brain activity? Again, you know, I'm asking questions because I'm an outsider. And so he said, you know, you're right. We have to rethink this, but it's going to take a really long time to get anyone to agree on this, because this is the way we do things. Well, we have to start changing the way we do things if it doesn't work that well. So what we did, Angela and I, what we did was we did a lot of things to this. First of all, we lined up, you know, these are design ideas. We lined up the labels. They're not diagonally, you know, we colored the aging, we colored the Alzheimer's, and yes, we kept the same colors because he wouldn't let me completely change colors. And then eventually we overlaid them. Because if you want to make contrasts, we think that this is a better scheme. But again, it's all about conversation. I'm going to jump ahead because I don't want to, you know, to keep you crazy. This is something else. But here, let me just quickly tell you this. Okay, this is from Klaus Hansons Lab. So these are two images, actually, which I made of a device. This is one image, and this is another image. And it's so long that you can't really show it in one picture. So what they did was they put it together, and they kind of, like in architecture, when you want to show a long corridor, you sort of clip it this way. And they labeled it this with this and this and this and this. And the first thing we're suggesting is, please give me more space around your picture. I mean, it's just too tight. So this is our idea. The next idea is, look at the way they labeled this, to, in our opinion, actually it's pretty much my opinion, get rid of the caps. It's why, again, simplify the image. Why not use lowercase? Get rid of the arrows, line up all your labels, and just use a line. Again, what we're doing is suggesting that any graphic element is distracting from what you want to see. There's one more issue in this image that we suggested is actually an error. Does anyone see something in the image that isn't quite right? Yeah, oh. Right. The insets, the SEMs, which, by the way, I'm not sure really gives any information, are in the wrong direction. They're not oriented in the direction of the full image. So we're suggesting, if you insist on showing that you've made an SEM, then at least have it facing the same way. Yes, are we going crazy with little things? Yes, we are. But I think it's worth it. It's worth it for those people reading it, and it's worth it for us to be more careful and pay more attention. Edit. That's what we're trying to get people to do more than anything. Okay, I really do believe that taking people through the story of how we think engages them in our work. So in the book, we have stories of how Rick and Carrie started working with sketches. They got this amazing artist, this is mitosis, to do her thing, which in my opinion is stunningly beautiful. And this, to me, should be the final images. They eventually wanted to computerize the image. And maybe I'm showing my age, but I think there's something incredible about hand drawing. It shows, by the way, it shows that it's a representation. There's a subtle distinction of seeing a computer-generated image and a hand-drawn. The hand-drawn shows that you're in process of thinking about it. I think that we need to use more hand-drawn. And this is a story about showing dark matter. This is how going from one to the next, they finally wound up with this. Interactive graphics is in the book. Not deeply, but this is probably pretty much what you all do. Is Kagate here? He was here yesterday, I think. This is really clever of how you could, using Google Map for the brain, clicking on a particular part of the map. These are spreads from the book. It's about how he was thinking about how to do this. You know Mariah's work? She tells the story of Mariah Meyer, how she went through all this. Ultimately, it is about collaboration. This is the extra piece that I stuck in. In Image and Meaning, which Bang discussed, it started in 2001. This is from 2005. We talked about 2D, 4D, ND, how far can you go, visual thinking in complex systems. This was about getting biologists, chemists, physicists, writers, artists, graphic artists to talk to each other. We became very involved with workshops. It was fabulous. We went around the country. The key is making it interdisciplinary. We can't just talk to each other. You have to talk to physicists who don't even look at your stuff. It's a fresh eye. I am absolutely convinced that bringing in fresh eyes is a very, very valuable thing. And I am going to just run through these. These are about collaborations going from what I think is, one thing that the researcher did. This is what I suggested that she do. This is from a lab of a figure. And the first question I asked is, what do you want me to look at? And it turns out what they wanted me to look at was this red area and how it connected with all this. And I said, hey, simplify, get rid of the pieces that really don't say anything. And if in fact that's all you want us to look at, then help me look at it. Heather is a student who had to produce a presentation at MIT, and I suggested Heather. Let's simplify it and get rid of some of those. I didn't say, I was nice about it. But I think there's a huge difference. And we collaborated on this. We collaborated on posters. This is a typical physics poster, which is unreadable in my opinion. So we go through this in groups. And so, for example, I took a panel from that poster. That's the way it appeared. And I suggested doing the following. First of all, I gave it more space, got rid of the ugly blue underlined writing, making those comparisons small suggestions, but I believe powerful suggestions. Adam is a fabulous student at MIT who really is eager to get his graphics better. This is a poster he made, and with collaborating in class, just by a few little things, made it more readable. Here's another typical poster in this world, and I just have no clue, what the hell's going on. And so what I did with the class as a collaborative effort was I first of all got rid of the text, just beginning, and then here's an idea that I'm throwing out at the kids. How about creating a poster that has an area like an abstract, a visual abstract, where you actually take the significant images so that when you're far away, here's another example. Sorry. When you're far away, which you tend to be at a huge conference, you see these. And maybe that would engage you enough. I mean, this is a whole different poster design. And I'm having trouble talking them into it because, it's different. But we could do that. We could start thinking differently. We talk about abstraction. This is an abstraction of polymer coming out of one of the labs from the kids, and we collaborated, and I asked questions. What in the world is this? What is this reflection on what? And so it turns out that this is more what he thinks the structure is. We could have a whole seminar on abstracting form. I'm sure you would discuss it among each other. But it's like night and day, but this is more of what he thinks it is. And these are just processes and how we talk about other things outside of science. Actually, this is fairly interesting. This is Occupy the Internet. This is juxtaposing information about occupying Wall Street and what happened on the Internet. This is from the Times during that whole effort. So ending finally is it's all about questions. And we, as a group, should be talking with each other about looking at each other's work and saying, what's wrong with this picture? Where does the metaphor fall apart? How do we represent uncertainty? I'm trying to get a seminar going next year. How do we represent uncertainty? How do we represent probability? What's the difference? and a ton of other questions should always be part of our collaborative community. I'm happy to hear from you. Thank you. Sorry about the glitch.